Good afternoon. It's my pleasure to welcome you to the Department of Radiology and Imaging Sciences Grand Rounds lecture by Dr. Steve Dager. Dr. Dager is a professor of radiology and an adjunct professor of bioengineering at the University of Washington, as well as an adjunct professor of psychiatry here at the University of Utah. He's the associate director of the Center on Human Development and Disability at the University of Washington. At a national level, Dr. Dager chairs the Department of Defense Congressional Mandated Medical Research Program's Vision and Integration Panel for Autism Research. He is also on the Scientific Advisory Board for Autism Speaks. Dr. Dager obtained his undergraduate and medical degrees from the University of Nebraska and did his residency at the University in Psychiatry at the University of Washington. He is a diplomat of the American Board of Psychiatry and Neurology and a member of the American College of, here we go, neuropsychopharmacology. Okay. His primary research has been the development and application of novel imaging techniques to study neuropsychiatric disorders affecting both adults and children. And during today's presentation, he will share his insights on the latest advances for autism spectrum disorders. So please join me in welcoming Dr. Dager to stage rounds.
Some of that's in past tense, but no longer chair DOD, which I'm very happy about because they'd always start the meeting Seattle time at 4 in the morning. So kind of like torture. So can everybody hear me okay? My head's stuffed up from I think being at high altitude because Seattle's at sea level. So I sound like I'm ringing in my ears, but and I can control this mic just by a button I can push.
It's the keyboard down below, right there. Yeah, yeah.
So this is where I hang out. Let's see. My lab, I'm very fortunate. I have a lab right on the Monterey Cut with the water. So I can sit there at night and write grants and watch the poutine row and all that. And they let me bring my dog in, which is really nice. So it I didn't have to even claim he's a therapy dog. We have lots of grants that support the work we're doing. I'm a consultant on Stephen Posse. He's an NMR physicist. We kind of started working with him at the NIH and then Germany and now in New Mexico. And I don't have any speaker disclosures. I do have a disclosure, as Donna said. I'm actually trained as a psychiatrist, so don't believe anything I say. But I love, I don't know, my research brought me to imaging early on, and I just love that I can apply imaging technology and help evolve it to study disorders that affect the brain. Do I have a pointer?
We'll get you one.
If you can, that would be great. So I mean, does everybody in this room know something about autism? Yes. Good. I understand. Yeah, with Janet. Yes, congratulations, by the way. We'll talk about that later. Okay, well, yeah, I mean, it's so that's actually wonderful news. And part of what I'll be presenting is we have a Autism Center of Excellence project that's been ongoing. So autism is a syndrome. And I don't know, I don't like syndromes, but that's part of why I study autism, to try to come up with more definitive.
It's a neurodiversity.
It's a well, it's gotten more of a neurodiversity controversy because clever people, I claim they're not, decided to lump together all sorts of somewhat separate disorders. So the first set of data I'm going to be talking about is studying people with autism disorder, which is a pretty severe defined neurodevelopmental disorder. But now it's been lumped together with particularly Asperger's disorder, which is kind of, it's more of mean, it kind of merges into eccentric personality qualities. And well, yeah, or Bill Gates. We get some funding from Microsoft, but I mean, and I've never diagnosed Bill Gates, so take that with a grain of salt. But and so as a result, there's a whole lot of controversy, no diversity kind of issues about, you know, this idea of quote curing autism. So, you know, somebody that has Asperger's doesn't want to be cured. But people with severe autism have a lot of problems, and that's what we focus on in terms of how can we make their lives better, and that's what most of my research is focused on. So they all have social impairments, repetitive restricted behaviors, and problems with social communication. They may have language disorders, cognitive impairment, and seizures as part of their disorder. I'm going to move on quickly because I want to get to our more recent stuff. Oh, thanks. I'm hyperactive, so I like having a baton that I can, yes, so thanks. But you know, autism merges, spectrum disorder merges with lots of other childhood disorders. So you have obsessive compulsive disorder, social anxiety. We've been studying kids since they were 3 months of age, and they're now going into teen, and we just got renewal to study them as teenagers. And you start seeing all these issues of social anxiety, aggression, and that as the kids age is what we're now starting to look at. Kind of the whole impact of raging hormones on this neurodevelopmental process. And ADHD. As Donna said, I used to chair, but what we did was we would decide on funding focuses with the Department of Defense. And I would share lots and lots about GI problems. It seems like all kids with autism have GI problems, and the parents really focus on that. Sleep disturbances are a big problem. I'll just mention in passing one paper we just published on sleep disorder and brain development. One of my colleagues just got a 5-year grant to kind of start to study the lymphatic system and you know how that might relate to neural development, which I won't talk about today, but it's kind of cool. So the first study I want to talk about is the University of Washington early development study. And this is how I got pulled into autism research. I had helped develop a chemical imaging technique called Pepsi, which some of you heard about earlier, sorry. And the overall PI, who was on actually a study section that rejected our grant to try to get it working because they thought it was crazy, was like so amazed that it all worked, and we published it in Fancy Physics journals and stuff, that she asked me to head up the imaging part of a program project grant. And initially was at 3 years, and then we kept getting renewals to look at 6 and 9 years, looking at brain development in kids with severe autism, already diagnosed, as opposed to the work I'll talk about later. Very innovative. This is, by the way, a tongue-in-cheek slide. If I have a pointer, some of you actually, Donna knows Cecil Hayes. Cecil Hayes was a coil. He made the first quadrature head coil for GE for the Cigna, and then got hired away to Seattle. And he used to make phased array coils with me and fluorine 19 spectroscopy coils. And this was a coil he made especially for our first PED study. And I just wanted to point out the glue sticks that we used to help. The glue was not in them anymore, but we actually used them to kind of ramp down and kind of help keep our kids' heads. And this was a linear birdcage coil. Let's see, I talked ad nauseum about propofol anesthesia in the earlier. The main thing that we did that I think was like really innovative is I had been doing studies dynamically. We would do something and look at brain metabolic changes. And I said, if we're going to study kids with autism, we need to study them longitudinally. And at the time that I got in the field, almost all the studies lumped together individuals with autism from age 3 to adulthood. You know, in a study, you know, looking at control that not even age matched necessarily, but they were lumping together across a whole developmental time period. And as I'll show you, that was really crazy. I mean, it doesn't make any sense to lump together different developmental periods. And so we were very fortunate that it was a longitudinal study. We had very discrete age ranges, 3 to 4, then they came back at 6 to 7, and then 9 to 10, and then they came back as teenagers. Let's see, that's all I want to talk about there. When I started, we had a GE Sigma scanner, 1.5. The gradients weren't capable of doing fMRI or DTI. And it's really funny, one of my closest collaborators now wrote the first paper on DTI, Bob McKinstry, as a postdoc. And we weren't able to do it at that time because we didn't have quite the equipment. But we were able to do spectroscopy. And we developed an approach to do, rather than single voxel 2D chemical imaging. My original work was single voxel at a 27 cc voxel size, which is not very anatomical, but we got it down to 1 cubic centimeter with our new approach, and ultra short echo time, which was useful for increasing our signal to noise and all that. So we get a real, rather than a single voxel in 4 to 5 minutes, we were getting about 1,000 voxels. So it was kind of cool, but it made a lot of work for my students to analyze it. The first thing that we found in this study, well, thank you very much.
Welcome, they replaced the battery.
Oh, thanks. Let's see, does that work? Uh-oh.
I'm seeing a light.
Try this one.
Well, I'll go on. I'll point, and it's anyway. The thing that we found, and why I emphasize that it's important to think about what you're studying, and in this case, to have a defined age range, is that at this 3 to 4 year time point, we found that kids with autism, with extreme autism, a fair amount of cognitive impairment, mostly nonverbal, that they had about a 10% cerebral enlargement. And this is the cerebrum without the ventricles. It wasn't hydrocephalus. Compared to our typical development, and much bigger brains in a group of non-autism, developmentally delayed kids. Yeah.
Just ask, did any of these include the single gene defect?
No, we didn't. Well,
Like
you could only, oh, thank you. You could only talk about what you measured. We didn't actually measure systematically. We did exclude kids with developmental delays that had like Down syndrome or known genetic syndromes.
Like MDX,
Yeah. Oh, yeah.
Or tuberous sclerosis.
Anything that was clearly not autism, we excluded, but we didn't do systematic genetic testing. But we definitely excluded kids with DD that had other, like particularly Down syndrome. I just wanted to point this out. We did a lot of different things with our structural images since we didn't have DTI or fMRI. And this is a study we did with colleagues at WashU, looking at the hippocampus. And we were interested, we did an eigenvector reconstruction. And the thing I wanted to point out is this inward deflection of the subiculum, which I thought was really cool. And unfortunately, the reviewers at AJNR did also, because what we knew was that this is associated with temporal lobe epilepsy in adults. And so the potential for a biomarker for seizure, fortunately, we didn't have enough kids that developed seizures to really prove it, but there seemed to be some. And the other problem was, as I'll get into later with our newer work, we weren't able to bring this down to the individual patient. So these are group differences, if that makes sense. So there were group differences in the reconstruction and word deformation, but we weren't able to really talk about the individual patients, which we can now with some of the stuff we're doing. This is my amygdala and brain. Colleagues in Seoul National University have worked with me for a number of years, and this was a MD PhD's dissertation project And they sent me for my birthday one time, my amygdala dancing. It was really kind of cute. And what we found was the lateral basal region of the amygdala was particularly affected in kids with autism, was enlarged. And we did some work, which actually isn't enough here in the references, looking at it as a prospective marker of outcome, and it was associated. The larger the amygdala, the lower the trajectory of social communication development, which we're now actually replicating with much better methods in our large multi-site study. So these are some of the things we found, and I'm really behind schedule, and I apologize. Bigger brains, there was specific subcortical involvement. My colleague Dennis Shaw wrote about a disproportionately smaller corpus callosum, and I'll show you some pretty pictures of that later on. But the main question was, as I emphasized at the outset, do these anatomical relationships stay stable? And that's kind of one of the themes of my talk is, I mean, could we have lumped together lots of different age range? And I would claim no. So this is a group, in red are kids with autism. Black is our typical developing kids, and the green were our developmentally delayed kids. And over time, and by the time they were 9, and definitely as teenagers, they're not on here as teenagers, the earlier 10% cerebral enlargement was no longer evident. And it's pretty impressive. And the kids with developmental delays, kept their smaller brains, the cerebral volumes, but there was a dynamic change in the other kids. And I'll show that also in some of our chemical data. So this early brain enlargement, I mean, one could say, wow, who cares? Which, I mean, you can't see it on an MRI. So you couldn't just do an MRI. I mean, lots of families ask me, well, is my child's brain abnormally enlarged? And it's all a Gaussian distribution, and they're shifted over. But it was one of the, at the time, the most replicated findings in young children, and I'll emphasize that. And these are some of the, so this was a paper we wrote, and these are some of the other sites. And we ended up actually developing our Autism Center of Excellence with colleagues at Chapel Hill, who had some of the similar findings. And so this is a famous science cover. Well, I think it's famous. Looking at genetic modeling approaches for autism in rats, or in mice, excuse me. And people were interested in B-catenin, but it doesn't look anything like a child with autism brain. It's actually pathological. They don't survive. And this was the point. It's not particularly illuminating to what we were looking at. So my interest was, so why do they have larger brains? And this is where maybe it'll be interesting, I hope, how one could creatively use imaging modalities to try to answer specific questions. And that's kind of what I like. So we were getting both SPGR T1s or SPGRs and proton dense T2s. And we were able to come up with an approach, if anyone's interested, the references on the next slide, to do quantitative T2 weighted measurements. And we were interested in, did they have bigger brains because they had a more accelerated normal development, if that makes sense, if they were just speeded up in terms of their development, like, their brain on steroids. And we wanted to measure the T2 relaxation. And that if it was an accelerated normal brain growth, we hypothesized a shortening of T2 relaxation. And we found the exact opposite. A lot of science is like you find what you don't expect. And what we found was that, as we predicted, the kids with developmental delays had prolonged T2 relaxation. But the ASD kids were also prolonged compared to the typically developing kids. So it was, if anything, more consistent with delayed brain maturation. not acceleration. And this is a paper in neurology if anyone's wants to look at it. So what did we do with our spectroscopy data? I cleverly, when we wrote our grant, had this hypothesis based on the limited literature of cerebral enlargement that there would be a abnormality of synaptic pruning, apoptotic processes, and that that there would be increased neuronal density or neurophil density. And we use spectroscopy. This is again is our 2D technique. Pretty pictures, wonderful spectra. I gotta say, it really works well. If anybody's interested in spectroscopy, choline, creatine, N-acetylaspartate. N-acetylaspartate. I became very friendly. I got on the editorial board of Archives of General Psychiatry, which is now JAMA. And the editor who knew me well was the one who really pioneered looking at N-acetylaspartate as a signaling, this is Joe Coyle, signaling mechanism for, why do cells want to elaborate and develop synaptic junctions and axonal elaborations and synapses. So we were very interested in NAA, both as a marker of neuronal density, but also signaling processes. And so we predicted, cleverly, increased NAA concentrations to go with the bigger brains and decrease metabolic relaxation times. I'm going to really push along, but this was our model. This is like more tightly packed neurons. These are little glial cells in here. And that there would be, because there wasn't enough space to evolve, that there would be a much shorter T2 relaxation in the more tightly packed cellular environment. We found exactly the opposite. So, so much for a priori hypotheses. And it wasn't as clear, but there was good evidence. This is actually gray-white matters. When we looked at regional, there's a neurology paper where we looked at ad nauseum regions and looked at the actual chemical relationships. This is showing the gray-white differences. So actually, exactly the opposite of what we had predicted. And the question again, would longitudinal relationships help illuminate what was going on? So this is work that I think you knew Neva in Seattle. She's still playing with me. What we looked at was, I'm just showing you the NEA. We looked at a number of different chemicals over time between 3 to 4 years and 9, and also into teenagership, and it was consistent in teen. And what we found, so the autism kids in red, and the DD kids in blue, and the typically developing in green here. And this is gray matter, and this is white matter, NEA. Particularly for gray matter, definitely the kids with autism had lower NEA levels at 3 to 4, and slightly lower even in the developmentally delayed kids. But by the time they were pre-adolescent, their levels were not distinguishable from our control subjects. A little less impressive in the white matter, but the same pattern. Whereas the developmentally delayed kids without autism, and I want to emphasize that, I had really good colleagues, clinical colleagues, that could, definitively say this is a child with developmental disorders, not autism. They stayed unchanged pretty much. And so some sort of consistent process. So anyway, so there seemed to be a dynamic brain chemical catch-up, and particularly for the kids with autism. And we didn't see that with the DD kids. And the one thing that we were interested in is that this is and Donna, you might be interested in this is consistent with chemical changes with injury recovery and traumatic brain injury. And so we thought that was really pretty cool. And it looked like, so there seemed to be some early event that seemed to I mean, the behavioral manifestations didn't go away. I mean, think of throwing a pebble in a pond. I mean, if you don't develop language at 6 to 12 months to 24 months, you're going to have lifetime, reverberations of that. But at least some of the underlying developmental processes we were measuring seemed to normalize over time. Hence, the sad thing, and this is full disclosure, I spent a lot of time looking at whether some of these chemical markers and changes could be prognostic. And I wasn't able to find it. Part of it was maybe power, but we really didn't find, that NEA didn't. So about half of our kids over the course of the study got much better. I was mentioning with the residents that one of them sent me his college graduation from Tufts. He was at the top of his class in computer sciences, you might wonder. But when he was young, we had to use propofol anesthesia to study him. And he was nonverbal and he became more verbal and much more interactive and excited about the research as he got older. But his NEA levels and changes didn't predict that. I just want to mention briefly, I was part of a NIH panel on mitochondrial disorders, and we got a lot of hate mail later on when we published our lactate data. I like lactate, and I started out my life measuring brain lactate, and we used it as a way to indirectly look at mitochondrial dysfunction in the brain. I will say that peripheral markers of mitochondrial status have nothing to do with the brain whatsoever. It has a lot to do with the genetics and different cell lines in the brain. So you could do a muscle biopsy and look at respiratory chain abnormalities that tell you nothing about the brain, which I would also claim is the same case with epigenetics right now. If you get saliva epigenetics, it doesn't necessarily tell you anything. So this is what we found. And in a nutshell, we didn't find anything to suggest mitochondrial dysfunction or respiratory chain changes or in our kids. And we looked every which way. I wrote it was controversial because, and still in some parts of the country, hyperbaric oxygen treatment was really popular and there was tremendous money to be made. And I ended up having to write a commentary on some of the people that were, beating the drum, had a vested proprietary interest in that they were profiting tremendously from putting kids and adults with autism into these hyperbaric chambers. And part of our commentary that did get me some hate mail was that if anything, putting them in an oxygen-rich environment would increase free radicals. And if there was a mitochondrial problem, that would actually potentially make it worse, theoretically, at least. So, so the bold is what we're actually kind of currently studying, which I want to move to, but I'm way behind. So the question was, is this enlargement at 3? I mean, clearly it was the tail end of something going on earlier, and but we couldn't diagnose autism earlier. So we were kind of at the point of where we could diagnose it and image things. And also with our mitochondrial findings, theoretically, you could certainly see something earlier on that you might not see later. And Donna's heard me ramble on about this in Seattle. We've been developing diffusion-weighted spectroscopic imaging techniques to try to indirectly measure inflammatory processes, looking at macromolecule tumbling and axons. And we've published some on that, but it's not ready for prime time yet, and I won't talk about that today. But that's one model of really, why we might be seeing some of these changes is inflammatory processes. If we were doing PET studies on them, we could use.
Those are not, those tracers are not without issue.
Yeah,
They still
have problems. Yeah. Well, no, I didn't put it in here finally. There's a couple of interesting papers on early microglial activation and autism that I think are interesting. A paradigm shift was a colleague, Sally Ozanoff, published this paper about if you have a sibling with autism, you have a much higher risk of having subsequent children developing autism, about a 20-fold. And that's what we found in the study I'm going to be talking about now, actually a little bit higher, 25%. So most of the kids don't develop autism, although they may have some symptoms on the spectrum, which I won't get into this greater, umbrella of autism. Yeah, the whole broader phenotype, but in terms of really severe diagnostic criteria, about 25%, 23%, I think it was, of our babies developed autism, diagnosed at 2 or confirmed later in age. And even with that, I mean, you can't image hundreds of babies to try to find, 20 or 30 that develop autism. And so this became a network study. See, in Utah's here, because I've been scanning lots of babies in Utah over the last years. Actually, it stopped during the pandemic, but one Memorial Day in the middle of the pandemic, I came out here and scanned a bunch of. It was my first flight during the pandemic. It was kind of exciting. So we're doing the recruiting from this part of the country. Also, some kids have come down from Alberta. Washington University, led up by Robert McKinstry and Kelly Botteron, who are very involved with the normal SIBS brain development study. And actually, I was having drinks with his wife and Bob one night, and embarrassed Bob, but it turns out as a med student at Harvard and MIT, he was on the very first ever fMRI study with Bellevue, and he wrote the first PTI paper as a fellow at WashU. And I had to explain to him that our scanner wasn't up to it to do those studies when I started out in this field. But anyway, and then Chapel Hill, which isn't labeled, it's supposed to be, North Carolina, Joe Piven, who's our overall PI of the network, and then Penn and CHOP in Philadelphia. And we've added on for some more recent studies, Minnesota, and some of the people at the High Magnet Center. Again, the main thing, this is a mom, actually a grad student, who, this is when she came back at 12 months, but I was walking her back the parking lot, and she had come in the middle of a blizzard, which is rare for Seattle. And I was walking her back, and she went over to the bike rack. And I was like, "You biked in the middle of the snowstorm?" Yeah. And she said, "Yeah." And I said, "You can't do that." She said, "Well, you wanted me to come in." She didn't have a car. But anyway, so here's her baby. I don't know if you can see her baby. It's really cute. See her little face here? And she came back at 12 months and 24 months, biking her kid in the middle of the night. These are studies done late at night. That was funny. Actually, at UCare, we have a Lego Phantom that for people who can't afford American College of Radiology Phantom, you can make them out of Legos, which we did. And Legos are highly engineered. So
I know one.
yes, okay. Maybe, well, you could use the one we have at UCare. It's.
I actually made one of two flip flops.
Oh, that's funny. That's great. They're very effective. I mean, you could get, as opposed to the ACR one, you could get a 3D read of any structural distortion. So it's actually more useful than some of the other current phantoms. And we also had brains from Montreal Neurological Institute is where our DCC was, flying around all the time. So we've had various people from Montreal here at UCare also getting their brain scanned. And Bob McKinstry helped me buy a 32 channel head coil for this study that I'm going to be talking about. And I sent him this picture and he said, his quote was, "There's some things you just can't erase from your brain." He's never forgiven me for that, but they're really tight. So let's see. I think I promised Donna I'd show her one of my clocks. I rebuild old clocks. That's my passion outside of research. So this is about maybe a almost 300-year-old clock that still runs. But the whole thing is that timing's critical. I'm just going to show you another. This is something that came out in JAMA Psychiatry. If anybody wants it, I could send them a PDF of it. It's looking at, so you could diagnose autism when kids become verbal. And when they start to develop language or don't develop it. That kind of is a lot of what is used in the interactions. We've gotten it down, again, it's a kind of almost a Gaussian curve. Some kids you could diagnose really early, but most kids you could diagnose by 24 months now. Some it takes a little bit longer, but there's this whole, we call it prodromal because it's pre-language period with a lot of subtle, soft neurological signs. So both motor differences, which we're actually looking at with a kind of a video capture approach now, looking for non-invasive ways of diagnosing problems early. That then merges into what we think of as the core symptoms of autism. The thing I wanted to emphasize and what motivates a lot of the research, other than my liking why questions, is the mean age of diagnosis around the country, supposedly with our vaunted healthcare system, is a couple of years after. I mean, it would be like waiting to develop or to diagnose, meningioma, 3 or 4 years after someone started having, severe headaches or something. Yeah. So it's kind of shocking, but that has to do with resources and a lot of it has to do with pediatricians want to reassure parents, especially young parents, and it'll go away, they'll outgrow it, and sometimes it does. But as a result, the horses are kind of out of the stable by the time it's often diagnosed in terms of of critical periods of language development and social interactions. And that's what we're trying to, I'll get to that at the very end if I have time. And again, this is a great quote I like. Development itself is the key to understanding developmental disorders. And that's kind of what we're all about with this research. And these are some of the critical things that happen in that 6 to 12 month that we're focusing on. You get joint attention where, baby kind of works with their parent to look at something together, and you have, they start to get face identification. This is during the pandemic, we had a lot of problem, even when we were back to scanning babies, because we were wearing masks and the moms were wearing masks. And I had to work hard to be able to get permission for the moms not to wear masks. At the time we were allowed, we were the first set of studies during the pandemic in Seattle because babies were thought not to be vulnerable to COVID at that time. And they, and we were doing a marijuana study where moms were pregnant during the first trimester, and if we didn't scan them, that 9 months of effort would be gone. So, but we had to wear masks, and it really affected our success rates of night studies because they couldn't read faces, and that's such a critical thing during this period. And when we were able to let parents take off the mask and comfort their babies at the scanner, then we got tremendously good results again. And just everything that you think about as being a human, is kind of developing and emerging during this period. So I'll leave it at that. This is our study design. I'm going to move on just because I'm way out of time. We now just Tooth Fairy. We're actually working with somebody at Sinai. Who we collect deciduous teeth from our families. And this guy at Sinai is doing like a tree core and claims, I believe him, that he could actually look at different periods of development, looking at things like heavy metals, plasticizers, anti-inflammants, I can't talk. And actually look, exposure during certainly second trimester, third trimester, and then postpartum at different points. So we're involved in this tooth fairy study. And we just, whoops, well, I could use this to do it too. How fun. We're now looking at sleep disorders, and we just got renewed. We're following the kids are now coming in at school age. And we're just about finished scanning them. And now they're going to be coming back as adolescents. And I got to say, it's a family tradition for these kids. I mean, they come in, even if they have autism, they're excited about being part of this. And I have wonderful people that work with me to do the behavioral thing to get, preteens in the scanner. We'll see how it works as teenagers. But we only really had one success, and that was here at UCare, a child with really severe autism who spent, and again, the school age were during normal hours, and he was running around UCare. Fortunately, nobody was there, like full bent out. And we got a T1, and that was it. And the parents in the school practice, they had him in one of those tents, those long plastic tents, and they did all sorts of things. And he came in 3 or 4 times, and so that was unfortunate. But usually we get pretty great success, like probably 98% success with this. But we have people who are really trained at doing behavioral interventions that work with the kids at the scanner while we're doing the studies. Things like, think of yourself as a statue. I always want to say, yeah, with the pigeons pooping on you, but I don't say that. And they're probably thinking they're right. Anyway, I'm going to move on. I apologize. I just want to say that our behavioral mess, my colleague Annette Estes, so this is kids with severe autism. This is all prospective. We didn't diagnose autism until they were older. So all this work I'm telling you now was essentially blinded because we didn't know the diagnosis at the outset. But the kids that went on to develop severe autism were really clearly divergent in their development. This is gross motor and visual reception. Kids, with more mild onset of autism, these are our typically developing kids, and these are the kids with familial risk for autism, high risk, but that didn't develop a full-blown autism. So but early behavior isn't sufficient to predict outcome at the individual level. Just like I was saying that, our brain enlargement or some of our chemical measures, and definitely our vector reproduction of the hippocampus. So at a group level, you could say that the kids that subsequently are diagnosed with autism have clear difficulties early on. Gesundheit. These are the so now we can do DTI and functional MRI and morphometry, and we've advanced. I got one of those wonderful Obama grants, ARRA, and we spiffed up Pepsi so we could do 3D. So rather than single voxel and then 2D, where we get 32 by 32 voxels in about 5 minutes, we now get 8 simultaneous slices. And our voxel resolution has gone down to 0.3 centimeters. So it's actually pretty incredible. And now we're actually added on water scanning during this acquisition so that we don't lose data if the kids wake up before we get our water, a non-suppressed water scan. This is a fancy paper that came out in Nature where we replicated this early increased cerebral volume hyperexpansion of the cortices in autism. Let's see, red is the kids diagnosed later with autism, and somewhere in there, I think green is is the kids on the spectrum or you know, at familiar risk, but not. And then the blue is our typically developing kids. And and we claimed with a reasonably good positive predictive value and negative predictive value with pretty good sensitivity specificity that on an individual level you could predict autism based on some of these findings. I'm gonna and the main thing is that the early brain overgrowth is still seen here. In this, we did all sorts of machine learning. But this, we were looking both at cortical thickness and surface area in this work. And some areas were more hyper expanded, particularly the visual areas. And I don't know why. If anyone wants to say, well, why the visual areas? I don't know. But anyway, that's what we reported. And recently we just published a paper. I'm going to show this one just because it was kind of fun. And one of my colleagues just got a 5-year grant out of this. That early sleep problems actually were able to predict pretty nicely the volume trajectory of the hippocampus. In an inverse way that you might expect, early sleep problems were predictive of larger hippocampi. And again, we struggled with that. But we're now following up with this work at a much more in-depth. And again, sleep disorders are highly prevalent in infants who go on to develop autism. Do people, I don't know if anyone remembers Guido Garrick. Guido Garrick was here for a while. He moved out from Chapel Hill. In fact, I was being wined and dined at Log Haven one night, and Guido was there with a group recruiting him, and he was incredibly embarrassed because he hadn't really mentioned it to people at Chapel Hill. And he was like, "Hi, Steve." So anyway, he's now chair of bioengineering at NYU, and he's in charge of all of our DTI work. Isn't that pretty? Looking at maturation. What we found in a nutshell is that kids with a diagnosis of autism in the red actually had higher FA values at 6 months and also at 3 months, but that it was significant decrease by the time they were 24 months. And this just shows. I guess what I want to say is that it was in most of the major tracks, 12 out of 15 that we measured. So it went from increased to decreased, which we thought was really interesting. I'm going to buzz over the fMRI. I apologize. This is Robert Emerson at WashU. So WashU published all those papers saying that most functional connectivity studies are crap because no one deals with the motion artifact 3-dimensionally and they don't get enough data. So we're incredibly careful because WashU colleagues are our colleagues, but some of them were involved with this, the paper by Powers et al. that basically claimed that most studies could be ignored. So we're very careful getting tons of fMRI resting data and incredibly, we use this software called FIRM to look 3-dimensional torquing of the brain while they're and we'll get up to 20 minutes of fMRI data in sleeping babies. So and again, the paper that we published in Science Translational Medicine, we found really good positive, negative predictive value and really good sensitivity specificity. And for any number of different networks could be related to particular. We're actually replicating this work now. We're doing a whole new cohort of infants. So I've been scanning tons of infants over the last, post-pandemic, we just went into high gear. So I'm always sleep deprived, but we're seeing if we could actually use some of these network differences essentially reduce global and local efficiency, to predict individual problems as kids get older in terms of specifically domains, because here's how you get. So this is just, I'm not going to talk. I'm going to move on because I love Pepsi because it's dear to my heart, but I won't talk about it anymore. I just want to say we go through a tremendous process of co-registering onto atlases we developed at each age range, so 6, 12, 24 months. And that we define the voxels, these 0.3 cubic centimeter voxels within regions. And then we essentially combine them for a super FID across the voxels, which gives us tremendously good signal to noise. And it gets around picking and choosing voxels that you like within the region, a three-dimensional region. And having 3D data, to really define the 3D region is. So we have about 32 regions that we define anatomically in the work that this work was acquired on a trio though.
Okay.
And what was it was 12 coil head coil. And this is just our super FID. You can see all the spectra that go into making it when we fit it. We use a technique, LC model, that was developed by somebody in Boston, who I always thought was German, but it turns out he was a draft debater, went to Germany during the Vietnam War, then came back. And he's completely retired, and this $10,000 program is now free to anybody that wants to get into spectroscopy. So to me, the state-of-the-art fitting technique is, with no strings attached, available to researchers. And the fellow's name Covinger, or not Covinger, gee, my brain is dead. Provinger, Steve Provinger, and it's LC model. If anyone is interested, I could help you find it if you need it. This is just, we do lots of things looking at our spectra, including Kramer row lower bounds and all that kind of stuff. This is a paper we published in MagRes Imaging, showing that at ultra short now, we're using 11 millisecond echo time, so TE. We're able to get wonderful glutamate peaks without any kind of J resolved kind of imaging approaches. And so that's kind of okay. So we're actually frantically before I retire, trying to finish up with some of our chemical measurements. One of the things we're really interested in is doing machine learning approaches, looking at glutamate NEA interactions. So we're really interested is, as glutamate goes up, NEA seems to go down, and looking at across regions and across time at this relationship. And we're also looking at lactate right now. I just wanted to end with this before my acknowledgement and thank you slide. It's controversial now with the inclusion of individuals with Asperger and such. There's a lot of strong feelings in the neurodiversity community. If can we call it disorder? Do we want to treat it? And I think our whole approach is to try to come up with measures that would allow families to do their utmost to enrich their child's learning process and development to maximize their potential with this. We just published a paper on some of this stuff in one of the ethics journals, bioethics journal. But it's how do you make a human being's life better in a very difficult world, and particularly when aging parents, because one thing that is clear that older parents tend to have, particularly older fathers, most diseases of childhood are all blamed on the mother, and actually Bettelheim, who just passed away a few years, was a psychoanalyst who claimed it was all because mothers were refrigerators and didn't respond to their kids. I know, but he's dead. So don't speak bad of the dead. Clearly it's a biological process and we're still trying to learn what that is. And how do you, if parts of development go awry, how can you help compensate for that? These are my many colleagues. This is Guido Garrick, who used to be here. That's me. Bob McKinstry is there. My closest collaborator, who's Satoshi and Yoshimi's neighbor, in their house is right here. Annette Estes. The work I presented earlier was these individuals were instrumental. Alan Archer in anesthesia, who did all of our propofol infusions. Wonderful that he did it. And Todd Richards, who did his postdoc here with Dennis Parker. Before Trun did his postdoc here, or his grad students studies here. I don't think they overlapped. Some of my colleagues, Stefan Posse, who I worked with over many years, developing different versions of Pepsi, and colleagues in Korea. June Kim did her MD PhD, measuring my amygdala among all our kids' amygdalas and doing 3D. The amygdalas are blobs. We had no problems defining three-dimensionally reconstructed the hippocampus, but it was quite a major feat. Then we had to do an incredible amount of modeling to show that we were actually able to define the amygdala blob. I really want to thank my University of Utah colleagues. I started actually scanning here. Satoshi was made aware of it when he was interviewing here. Satoshi, as you might all know, Dr. Manishima came from the University of Washington. We worked not together, but he was my section chief. And he called me up and said, and I think he was in Japan at the time, and said, "What are you doing in Utah?" And I said, "I'm scanning babies." But Bill McNann, who was a former chair of psychiatry, helped get me going. And Debbie Bilder, who's a developmental psychiatrist, and also in pediatrics. Actually, if anyone's using UCare and you see a wood rocking chair there, that's from her. She donated it to me in our early baby studies so parents can rock their babies. There's a little bit of metal in it so we don't get too close to the scanner. And Perry Renshaw, I don't know, do people know Dennis Parker here? Dennis knew me from a long time ago. And so when I had this audacious idea before Satoshi showed up on the scene that I wanted to use their scanner to scan babies, he said, "Go for it." Wonderful man. But he was there one time early in the morning before we were coming on with a family, trying to get data for our ISMRM meeting. And I came in carrying this 32 channel head coil. And he looked at me like in horror. And he said, "That's Perry Renshaw's coil." And I said, "Yeah." He says, "He doesn't let anyone use it." I said, "Oh, he gave me permission to use it." And he looked at me, he said, "He wouldn't let me use it." So anyway, if he asked. Yeah, I know, Perry's wonderfully. But anyway, we had a 32 channel head coil in Seattle, but I didn't want to transfer it back and forth. And he had an extra one from this ABCD study. And he's been wonderfully generous. I knew Perry when he was a resident in Boston, so we go way back. So anyway, I cleverly took up all the time, but if there's any questions, I would love to answer them. I thank you really very much for your kind attention. I hope it was at least interesting. So thank you. Oh, turn on the light, yeah. Oh, come on, there's gotta be one question. Like, why the heck do we want to scan babies? I don't know. It's a lot of, I was telling the residents the hour before, what?
We have a longitudinal study. that
Yeah, I know.
Later, so if you want to know about teenagers. There they are.
I have a question for you about your functional scan.
Yeah,
All the babies, I mean, a lot of them have sleep problems too.
right, yeah.
How do you know that you're looking at sleep functional connectivity or? autism or,
That's a,
you know,
that's a great question. I've had those arguments. I mean, if the babies are awake, so we've actually scanned some awake babies who just are really passive, and we can't use the functional imaging data because, but the reality is we don't know if they're in REM sleep or they're, we had actually at one point debated putting on electrodes to at least get some EEG measures, and these babies, we didn't. So what stage of sleep they're in, we don't know. And we know that for our school age studies, they're watching a video throughout most of the scanning, but when we switch to the functional MRI resting state, we have them look at a crosshair. And if we have any evidence like their eyes closing, which you can see on the images as they come up, we actually don't use that data. And then one of my behavioral analysts who goes in there and pinches a toe or something says, "Hey, wake up." And so we throw out those, but you're absolutely right. I mean, we don't know what stage of sleep. One of my colleagues just got, it's a sonar kind of technique that claims, oh, a second, that claims for older kids that are more than 80 pounds. So they're doing this study in somewhat older kids. You can actually get an indirect measure of REM and non-REM sleep. And somehow it picks up the eye blinking. And so we're doing a bunch of, we're using actigraphy and different things to look at sleep status and sleep quality. But there was a huge fight with the study section who wanted us to do sleep lab studies, have them come in and hook up, and it was quite a fight we had to get this. Well, it was a fight, first of all, to get it funded, and then it got hung up for a year because our DCC site, the site that collects our data, was in Montreal, so the Montreal Neurological Institute, and the State Department had to sign off on it. And I'm sure if it had been any of a number of other countries, my colleague Stefan Posse put in a grant with that I was on with collaborators in Denmark, who had a combined PET MRI scanner, and we wanted to do some PET corollaries for neurosurgical approaches, and they would not fund the grant because it was, part of it was in Denmark. So they barely let us do it with our DCC in Canada, but it and the problem with, if you go into a sleep lab, these are kids who have real problems with wanting to maintain similarity. They don't like things in their environment to change. And if you bring them into a sleep lab, what are you measuring? It's so we look at it as naturalistically as possible. We're hoping to at least get a measure of REM, non-REM, which is not ideal, but it's certainly better than looking at movement as a measure. Yeah.
How long were the MR scans? Were they short? Did you have to shorten them? And how did you determine how long to scan the sleeping baby?
We argue about that. We just are going to get a grant funded as of today. We got just in time to study babies who were exposed in the first trimester to marijuana. We're doing it on a Philips scanner. And we were like just ruthless in cutting out things to make sure the scan was 45 minutes. For these scans, and I got to say the Siemens scanner is quieter. It has a little bit better gradients. I was talking to the residents about having to engineer the DTI pulse sequence on the Philips because it had to refresh, and you go and then stop, and then and they would invariably wake up after the first round. So we try to the group at WashU is great at
fidgeting with our sequences and to try to minimize noise. And we're going to be, we're dreading, but we're upgrading to XA platform. And you could do a lot more fancy imaging techniques, but they're really loud, so we're not going to make use of a lot of the increased capacity. So our sequences for the infants, well, they're about a little over an hour. And these are in sleeping infants. We don't always get everything. We did a study Monday night and we got through 2 shells of the DTI and they woke up and I said, we're done. And if we're fortunate at the end of that, we're trying to do some work looking at CSF flow. And usually when I try to put the pulse ox, we gate it to cardiac cycle, and you can imagine babies have really fast heart rates. So there's a different kind of hand waving than sleeping. But usually when I try to get the sleeve on their big toe, they wake up. So we're kind of failures at that. With the adolescent studies, it's a little bit over an hour also. We thought about adding a task fMRI study during adolescence, and we decided not to. And long involved arguments about that I could share, I guess. But yeah, so we're doing just awake functional connectivity. But thanks. Any other questions? I think we're done now, right?
Yeah.
Thank you again for your very kind attention.